I'm Ethan. Uh, so I'm in Lenore, so I know exactly where Hickory is because I grew up in the shadow of App State. So very familiar with that geography. But I was raised there, and then I moved to Raleigh in 2013 to study chemical engineering at NC State. I concentrated in nanoscience. Um, I was involved in undergraduate research for 3 years. I studied bioreactor design, and in particular, challenges facing the ability of cells to live in bioreactors, so cell viability, and some of the harsh and turbulent conditions they experience there. So looking for ways to protect cells in that regard. For a couple of summers, I worked in industry at a pharmaceutical company called Excella, which is also in that county. So if any of you are looking for a pharma company to work for near Boone, there was one option. But I worked there as an organic chemist for a couple of summers, making cart medicine. So that was really fun, but also challenging in a great way. And then one summer, I worked in South Carolina on REU program, which is a great thing for undergrads to do if they want. So I can talk about that if y'all want. But that was fun. I worked. That was my first exposure to environmental engineering because I worked on gas separation with membranes. So trying to capture carbon dioxide from effluent. I got rejected, kind of. No one ever got back to me, so it's like a soft rejection. But my resume was complimented, and so that fall after the REU, I sent an email to Mark Wisner, who had the lab at Duke, because he was the one who was running the REU, kind of. And so we met for coffee one day, and that's more or less how I transitioned into grad school after I graduated in NC State. So I've been at Duke since 2017, and I study bacteriophages, which are not vesicles, but they're on a similar size scale, and they're pretty interesting organisms. So bacteriophage, or sometimes called phage or phage for short. Bacteria, obviously it's referring to bacteria. Phage, it's the same root as the word esophagus. So bacteria eaters, which isn't precisely what they do, but it's kind of true. That's phenomenologically what it looked like they did whenever they were being first discovered. They look like they were eating bacteria, but they do infect bacteria. So a phage is a virus, but the host is a bacteria. And so this is not the greatest TEM picture of a T4 phage, but I'm proud of it because I took it. So you can kind of see the capsid, which is on the nose scale, kind of a classic size, about 100 nanometers. And you can see its tail coming off of it. It looks like it's contracted. But anyway, so that's a bacteriophage in a nutshell for you. It's a capsid, or at least this type of bacteriophage. They have different morphologies, but this particular type of phage is kind of the classic head, tail with little whiskers on it. And the capsid contains genetic material. It can be DNA, RNA, double-stranded, single-stranded. Again, depends on what phage you're looking at. But this is also the same phage I use in my research. It's a pretty well-characterized phage by this point.
So do you like breed these? Like, how do you find these things? Like, you like harness them somehow, or like have like a little culture of them? Like, how do you find these things?
You find them anywhere. So the ones I use, I do have from a stock solution, but so in early research, I just went to the Durham Wastewater Center and got a sample of dirty water. Essentially, so if you go to the next slide, there's a diagram that'll help with this question. Yeah, so that's more or less a life cycle of a phage. It's not really living, but at the point. So they depend on a host organism to grow. So still mystery where the first phage kind of came from, but once phages existed, they basically insert their genetic material into their host's organism, which kind of captures its replicating facilities, and that host begins to create more phage until eventually that host lyses and more phages are released. And so you can find them. You can find a phage in almost any environment where you'd find its host. And so if you are looking at a certain sample of water, they'd expect to have a certain kind of bacteria. More than likely, you'll find its phage there with it because they will kind of coexist together. I guess there could be exceptions to So yeah, that's more or less how you harvest them. You kind of just introduce them to their host, and then it kind of takes over from there. But a reason why we should care about them in an environmental context is that, as you could imagine, they kind of have a dominant role in how they the dominant role in the population distributions of certain hosts. And so you can kind of see how that would kind of work its way out in the environment. But a lesser known impact that phages have on the environment is, I don't have a diagram for it here because it's kind of complicated. They basically redistribute nutrients. And so whenever a host lyses or bursts as a result of phage infection, all that cell debris becomes available organic matter for other organisms to use. So other bacteria could use that for their own growth and development. And then you can kind of move up trophic or levels in the food chain, trophic levels. And so viruses play a role in kind of redistributing the organic nutrients in the environment in a way that keeps things from basically just moving up trophic levels until you kind of starve certain environments of certain nutrients. And so it has a pretty significant role in like the health of environments in more ways than one. But my research, because I work in the same lab as Nick, and so we're asking somewhat similar questions, but I'm coming at it more from the phage perspective. And so my research is focused on how do we model these phage interactions and their transport. And some of the methods that I use or plan to be using are similar to those that you would probably hear later in future presentations that Nick would use for vesicles or EVs.